That's important for us. The last route is the other way, which is where the viruses come in via the caveolal pathways. This comes via a non-acid bath route, and this may give us an alternative way of getting to our nucleus and our cytosolic targets. So again, if we can target cell membrane receptors that come via here, we have an opportunity to get in the cell via the back door. Okay? Biological barriers and cellular barriers. So that's what we're looking for today. It's a big ask, and when we put this together, we were questioned why the proposal is so broad. And the answer is there's not one solution here. In fact, the solution is going to come from a very wide-ranging. It could come from microbiologists, it could come from cell biologists, protein chemists, polymer chemists. We're looking for all that kind of breadth so we can solve this question, because by being too specific, we're going to miss it, miss that opportunity. So what are we asking from you? We're putting our hands out. This is what we're looking for. We're looking for molecular and cellular biologists. The opportunity to look at the mechanism of this uptake and trafficking. We need some imaging ability because we want to know where in the cell our compounds are going. And bear in mind, I haven't said anything about what type of payload here. We could be talking about an oligo, we could be talking about a peptide, antibody fragment. It doesn't matter here because they all have the similar delivery issues. Okay? And we want to make sure, as I said, we generate our in vitro and our vivo models so that we're sure that we're going to get something in the clinic that's going to work. A lot of companies have failed because they've just built models in vitro that have no in vivo translatability, and that's important. Next, we're going to start wrapping it up in the fancy stuff. We want to have an option for formulation, for nanotechnology. Protein and nucleic acid chemistry. Can we change the basic chemistry of our proteins and oligos to make them more penetrant, more likely to be delivered in the right place? That's the simple sort of small molecule idea. Just change the structure of the molecule until you get the right properties. We can do that. We can do that without losing our biological activity. We've got a way of doing that. But then let's try and generate some novel carrier systems that we can wrap them up and get them through those barriers and deliver to the target where we want to go. And then let's try and make sure we can make these at the scale we're going to need to support clinical and preclinical samples. There's no point if we can only make these in milligram quantities, because we're never going to do a clinical trial for that. We're never going to be able to do that. It's got to be scalable up to clinical trial levels. Even if we never get there within this project, we need that scalability. Okay, what we're going to bring to the table, this is what Ekkehard and my company and other companies will bring. In terms of supporting the direct projects, we're going to supply the payloads. You won't have to make your own oligos, you won't have to generate your own antibodies. We can supply those. We have several dummy versions, we have several versions we can use without breaking any kind of confidentiality rules. This is just going to be payloads that we know work and we can work with. We're going to be able to test our carriers and our systems in the relevant in vitro systems, and then going on to the relevant pharmacology studies and in vivo studies, the disease models. We can bring those to the table. And then when we get up to the clinical stage, we can help with the clinical trials. Now, as I said, this may never come within the lifetime of this project, but we can help with it. And I think that's an aspiration for us to make sure that we're looking towards that. Because there's now opportunities to go fast to man, where we do short studies with microdosing, where we can actually get into man with very small amounts of tox cover, because we use very small doses. And as I said, support functions, project management, and statistical analysis, database capture. So that's what we'll bring to the table. Okay. I said it's going to be pre-competitive. The only way this is going to work is if we're not sitting down going, this is my property, and that's your property, and this is their property. It's got to be free and open. That means if you bring IP to the table, it's still your IP. But what we develop together is our IP. Everybody's working together. Everybody owns that IP. And then the companies have an opportunity to license it off the back of the project. The IMI will coordinate that. And Maria, you've got some more information on that if anybody wants that. But there will be no, this is ours and we're going to make it our own property. It's sharing. And the reason we're doing that is we're not touching any of our therapeutic agents here. None of our drugs are going to be used here. It's going to be dummy molecules. So we're going to be able to do academic research using our molecules without me worrying about what Ekkehard's looking at my drug and he's looking at my, and I'm looking at his. That's not going to happen because we can use prototypical molecules here. The basic research of cellular uptake is going to be the mechanisms and the endosomal escape mechanisms. We're going to have that licked. We're going to work out how that's going to happen, and that's going to allow us to do the nanotechnology and the carrier systems associated with it. So by doing the basic research, we can then work out how we're going to overcome those barriers. Okay? Not one drug in there at all basic pre-competitive nature. And again, I'll stress this again, it doesn't matter if it's an oligo or a peptide or a small protein. The challenges are going to be the same. So we can use one system to deliver all of these with slight tweaks to get to where we want to be. Okay? We had to put a slide in for what's in it for you. This is hopefully fairly obvious, but we will go through it anyway. You're going to directly benefit patients with your research. We will use what you develop, and we develop, to get our drugs to patients in unmet medical need. Undruggable targets, they can't get small molecules there, and we can't get our big molecules there because we can't deliver them. And this will allow us to go from bench to bedside. The work we do in this room, will start in this room, will result in drugs getting to patients in the clinic and making their lives better. And you also have the chance to be involved in the next generation of drugs. Small molecules are dead. Small molecules are generic, small molecules are 10 a penny, and the regulators and the payers are not wanting to buy them anymore because they're not that much better than the generic version they have already. This gives us the next level in. It gives us the opportunity as part of a European network, and it'd be nice to be a European network and not just waiting for the Americans to do it all, but have a European network with access to academic expertise, access to the pharma drug development capability, because we can do drug development quite easily, and then access to the IMI resources to coordinate the whole thing together. And what are we going to get out of it as pharma? We're going to get novel therapeutic opportunities. We're going to find ways of delivering our drugs which we didn't have before, because it's going to be a system that's developed, and we get to tap into a European-wide brains trust to help us do that. And again, we have the strengthening our new portfolios. This is going to improve the therapeutic index. We can dose less. We have to use less toxicity. We can reduce those doses. And we were discussing this over lunch. Right now, these drugs are the sort of the new kids on the block. But 2-3 years is a long time in industry if you don't have results. And pretty soon they're going to start losing interest. And some companies have pulled out the oligo field very recently and very publicly because it's not delivering for them. And that's because they can't get the delivery. If we can get the delivery, these drugs will become part of it. And it really finishes off, and it's a bit corny, but novel medicines plus improved delivery will give us better medicines. They're going to be medicines that get to the target, that have less side effects, that have less cost of goods because they're getting to where they need to be only, we're not wasting them elsewhere, and they will hit those unmet medical needs to make those patients do the business.